So I'm turning to our next speaker, Helena Hogberg. She is the Deputy Director of our Johns Hopkins Center for Alternatives to Animal Testing, and she holds a PhD in toxicology. And together with the CAAT Director, Thomas Hartung, she founded the CAAT Lab in 2010. And her research focuses on developmental neurotoxicity and the use of emerging tools such as 3D organotypic cell models, induced pluripotent stem cells, and omics approaches. And yeah, you will get an idea about Helena's work. And if you want to hear more from her and also see our virtual lab, like a virtual tour of our lab, we are actually going to have a summer school on innovative approaches in science. I thought this is a good forum to talk about this because it's a whole week. It's starting in 2.5 weeks. I will send the link in the chat. You can sign up for free. And if you want to do the whole course, you get a certificate. You can also sign up for just single lectures. And it's an international group of speakers, not only from Johns Hopkins, but also Harvard and EPA, NIH, and so on. So Helena, thank you so much for agreeing to do this. You have a busy schedule at the moment with so many speaking engagements, and I'm happy that we could win you for this 3R symposium. Please go ahead and share your slides.
Thank you, Catherine. Let's see. Can you see it?
Yes, looks good.
Perfect. Okay, thank you, Catherine. Thank you for inviting me also. It's a pleasure for me to be here and share some of our research. It's a little bit different audience than I'm used to, so I hope I will try to do it basic and not be too fast or try to capture too many details, because I think you don't have the same background and I'm not used to talk to this kind of crowd. But if you have any questions, any clarifications you need, please don't hesitate to ask. So what I'm going to talk about is our Minibrain Organoids model and what we have been using it for. So I want to start with a disclosure because I do have a patent file with Johns Hopkins University on this mini brain. And I'm also on the scientific advisor board for Axosim Inc., which is a company that are licensing our mini brain and providing services for pharmaceutical companies and others on this mini brain. So I do have some interest in showing good data. First, I want to acknowledge the team because this is not work only done by me. Here you can see our team, and of course, like in everybody else's cases, we have been on quarantine for a while. Since mid-March, we are slowly going to start to open our labs again, probably by the end of next week. So these are the people in our in our team, and we, as you can see, are still very active, but we meet remotely most of the time. Okay, so let's go into what I want to talk about. Why do we believe that we need to do some kind of replacement or alternatives to the animal approach? Well, the truth is that current in vivo approach does not necessarily predict human health effects. And this little cartoon is showing, this is for drug industry to develop new drugs, that we have some in vitro and in silico and some screening approaches where drugs are first have to pass. They go into the animal and they pass in the animal and then they go into clinical trials in humans. And unfortunately, most of the drugs are failing there. So 89% of drugs fail when they come to clinical trials in humans. And this is mainly due to safety or safety reasons, or that you cannot have the same kind of effects or efficacy in the human compared to in the animal model. In my case, I'm very interested in the brain. That's where I did my PhD. And when it comes to neurological drugs, it's actually even worse. It's 94% failure rate for neurological therapies. Some neurological disorders, such as Alzheimer's disease, for example, is extremely bad. We only have 99.6% failure rate. So it's very, very few drugs that seems to work in clinical trials for humans. And this, of course, leads to a very high cost, about $2.6 billion, and it takes a lot of time. So we feel that we could do something better. We need to do something better. We need to find different approaches that are better in predicting the human effect. So again, I'm interested in the brain. That's where I'm going. And there's a lot of in vitro systems available for this, monolayer cultures. But we are working with three-dimensional systems. And why do we think it's important for the neural systems or for looking at neurological disorders? Well, all organs are, of course, a challenge to model in vitro. They are not very simple, they are very complex, and that goes especially for the central nervous system and the brain. Because the brain is the most complex organ in the human body, it consists of numerous different cell types. It has different kind of neurons as well as glial cells, so astrocytes, oligodendrocytes, and microglia. They have very complex neural networks and brain cell architecture, and this is very difficult to capture in a monolayer culture. Cell-cell interactions is extremely important for the function of neurons or for the brain. So cell-cell interaction and biochemical signaling is crucial. And of course, if you have a three-dimensional structure, you have a much more higher cell-cell interactions. The 3D structure is also essential for some of the neural processes. So for example, myelination and synapse formation, it's impossible to model in a two-dimensional culture. So we have developed a three-dimensional brain model of a human brain model. We have had a three-dimensional rat model in the lab for a very long time. But we felt, of course, that there's a species difference that we know about already. So we felt that we need to develop a human brain human brain model. And we were fortunate and got funded by the NCAT NIH to do this. So the picture you can see of the 3 people here are Thomas Hartung, the PI, myself, a co-PI, and then David Pamies, which was a postdoc at that time, now faculty in Europe, in Switzerland, that has done most of the scientific work or the development of this model. But we were 3 partners, so it was us, CAAT, and as well as School of Medicine. And we teamed up with them because we did not have a lot of background in stem cells at that time, or induced pluripotent stem cells, which the School of Medicine did. So that's why we teamed up with them. We also teamed up with Kennedy Krieger Institute, and they're very good in developing certain assays and are also the leader in neurodevelopmental disorders. So again, the goal of this was to develop a human mini brain. So how are we doing that? As I said, we're using human-induced pluripotent stem cells. We are culture them first and differentiate them into embryonic bodies. Actually, we do now have a different protocol, which goes quite much faster. It goes directly from induced pluripotent stem cells to NPCs, neuroprecursor cells. But this is the original protocol, and all the data I'm showing is with this protocol. So once we have them into neuroprecursor cells, we are differentiating them in monolayer culture or proliferating them in monolayer culture until we get enough cells for making our mini brains. And then we are scraping them off manually, as you can see in this little cartoon, and we are putting them in 6-well plates on a shaker, a gyratory shaker, so that you can see here, very simple. And due to this moment, the cells are actually clustering together and they form this 3-dimensional brain, mini brain that you can see on the side. What does the model consist of? Well, it consists of all different cell types in the brain, or almost all the different cell types of the brain, so different kinds of neurons as well as glial cells. One glial cells that we're missing is the microglia, which is the immune cells of the nervous system, which is of course very important. However, we can add that one afterwards. The reason we don't have it is it's because it's differentiating from a different germ layer. So it needs to be added afterwards. The size of this mini brain is about 350 micrometer, and this is a relatively small size, which make sure that we have no necrotic or apoptotic cells in the core, because the nutrition and the oxygen reaches the core. Bigger the organoid gets, it gets a bit more complicated and more, you kind of need to have some kind of flow system that goes into the core then. What we have seen is that our model is very reproducible in size and cell composition. So it might not have the most fancy structure that some of the other models have, organoids have. So we kind of not even really calling our model organoid, we more call it a sphere. The reason we did not want to focus on getting the architecture was really reproducibility, because we wanted to be able to use this one for drug discovery or for toxicology. So we wanted to have a more of a throughput platform. And therefore, we need to have it complex, but still simple enough that that's possible. So genetic, so we also, as I said, we're using induced pluripotent stem cells, and therefore we can use different genetic background from patients, so different genetic background. Not only do we have present cells, they are also functional. So here I'm showing some functional assays for neurons. So the way neurons are communicating with each other is with electrical activity, so spontaneous electrical activity, which you can see here to the left. We are recording that with microelectrode arrays, so MEAs. So basically we're observing how the cells are communicating with each other. On the right, instead, you can see calcium imaging, and this is one brain sphere, and you can see, hopefully, that it's you can see a movement of the signal between different cells. So these are the small blobs and you can really see how the signal is going between the different cells. So they are really communicating somehow. We just have to figure out what they are talking about or what they are thinking about. Another assay we have in-house is to look at how the neurites are growing out, and this is especially important for neurodevelopment. So the cells are migrating, and as well the neurites are expanding. So this is what's happening in the brain during development, and we can use this as an endpoint to look at how different chemicals might affect migration and neurite outgrowth. And you can see here the control situation and then exposure to a pesticide, how the migration or the neurites is much smaller. So I show you now that the neurons are functioning, also the glial cells are actually functioning. So oligodendrocytes are myelinating, that is the role of oligodendrocytes. So what you can see here is staining, two different kinds of staining, myelin basic protein in green and O4, this marker for oligodendrocytes, and NF is a marker for neurons, neurofilament. What we have done here is we have developed an assay with staining that makes it possible for quantifying how much myelin we have. And this is something we are extremely proud of, because it's very few in vitro models that have shown de novo myelination. And myelination is a very important compartment in the neurons, because it's very often affected by chemicals, but also in certain diseases. So multiple sclerosis may be the most known one, but also ALS. And what in our assay, what we're doing is a 3D reconstruction, as you can see on the left. And on the lower picture of the left, you can see how it's really true that the myelin is wrapping around the axon. So you can see this with electron microscopy. When we are quantifying this, we can see that we have a massive increase. So at 8 weeks, we have more than 40% of myelinated axons. And to my knowledge, this is one of the few models where we really have this high myelination. So what is the application and the research we're doing with our model? Well, we started with toxicology because that's our background. So we do a lot of neurotoxicity or developmental neurotoxicity, but we're also going into looking at more disease, so neurodegenerative disorders, for example, Parkinson's and ALS. I said already, we're doing some demyelination. So here's, for example, some multiple sclerosis work, but also just looking at how we can remyelinate after we have a demyelination of the axons. We have used our model for cancer. We do some resilience work, so not only how the cells are dying, but also how they are coming back. We know that the brain is, of course, protected by a blood-brain barrier, and we're doing some blood-brain barrier work as well, because it's not sure that both drugs and toxicants are reaching the brain because of the blood-brain barriers. So that's, of course, an important work that we also are including. Another thing that we're doing, because we use induced pluripotent stem cells, we have different genetic background, as I said, and we can therefore study gene-environmental interactions. And was already mentioned by our keynote, gene-environmental interactions or environment is very important. And we know that certain backgrounds have higher risk factors of developing diseases, for example. Some example here where we do some work is autism as well as Down syndrome, where we know that the background makes them more vulnerable to certain exposures. Another thing we're doing is inflammation. I told you already we have included microglia to the cell model. And here we are looking at different infectious diseases, different viruses, so Zika, JC, HIV, dengue, and actually we're currently doing some SARS-CoV-2 research as well. Unfortunately, I cannot show you that data yet, but hopefully it will be published shortly. So what I will show you, so I felt that maybe toxicology is of less interest for you. I thought perhaps most interesting for you would be more the disease modeling that we're doing. So I wanted to show you some with the Zika virus. And Zika, as you probably were aware of, is spread by mosquitoes. So it's infected mosquitoes. It can be also transmitted from a mother to a fetus or through sex, and likely by bodily fluids. And the main thing that can happen during pregnancy, or the more severe pathogenesis, is that it can lead to microencephaly and other severe brain defects when it's spread from the mother to the fetus. And that's what you can see, microencephaly, in the little picture in the corner. But there are also evidence that adults can also get some problems with the immune system. So Guillain-Barré disorder, for example, where the immune system attacks part of the peripheral nervous system, is something that also have been linked to Zika, as well as other neurological conditions. And one of the issues with Zika and the virus in general is that it's hard to have good animal models that can be infected by Zika, and other viruses as well. As you know, most of these disorders are actually something that is just happening in humans and not necessarily in animals. If you want to have a good animal model for studying virus, and you probably know this more than me, but often you need to do an immune suppression of the animal. And again, we know that the immune system is important in the pathology in these disorders. So if you don't have an immune suppressed model, I'm just wondering how this is really mimicking the human situation. So as I mentioned, we had added the microglia to our cells. We felt it's important when you're studying viruses, but also for other things. So here you could just see that we successfully managed to incorporate microglia with different markers. So this is for 3 different markers with electron microscopy on the first upper one, and then with immunohistochemistry. And then we wanted to see if this also were functioning. So what we did was that we challenged the system with LPS, Lipopolysaccharide, so which is part of the gram-negative bacteria. And what happens when you add this to tissue or to humans, or you can see an increase in cytokine release. So what you can see here on the graph is first BS stands for brain sphere. So it's only our brain model without the microglia incorporated. In the middle, micro BS is a brain sphere model with the microglia. And then we have the microglia alone. And what you can see here, very interesting, is that only when we have the combination of microglia and the brain sphere, we can actually see an increase in cytokine. And this is done over a 24-hour period, and we know that the effect of the cytokine release is changing after infection. So we need to look at this window. And this is an indication for us that there is some kind of communication between the brain spheres, or the other cells in the brain, and the immune cells, the microglia. So we then also infected them with Zika. So we used 2 different strains of Zika that you can see here in the graph, is that we could see that the virus could replicate within the brain sphere model, which means that they have to go into the cells to be able to do this. That means that they have really been infected, and we could also see that on the staining in the lower picture. Again, we looked at cytokine release after exposure or after infection with Zika, and again we could see that when the microglia and the brain sphere were together, we actually saw a stronger effect, again indicating that the cell-cell interaction is very important. Finally, I want to show a few slides on glioblastoma. Glioblastoma was already mentioned earlier. It's the most common and most aggressive type of primary brain cancer, and it's very difficult to treat. The typical survival length after diagnosis is only 12 to 15 months, so this is a very sad disease. And when it comes to cancer drugs, we know that 90% fail. When they were doing something in the mouse model, they did not do anything in the human. So that is quite high for this. And we know that rat and mice only predict each other with 57% in the cancer bioassay. So therefore, what is the likelihood that the rodents would predict the human any better? And even if we have done some success in some cancers, that unfortunately does not happen with glioblastoma, it still doesn't have really an improved survival rate, even if we have succeeded in other forms of cancer. So what we did here with our mini brain model was that we incorporated a glioblastoma from patients into the brain sphere. And what you can see here to the left is how you probably, I wonder if can I show you with this one? Yeah, here you can see the tumor which in the microenvironment, and how it's growing over time, and this is shown here in this graph. So you can see we did not see a change in the amount of neurons, but we did see sorry, Sophia, you have to go. But we did see a growth of the tumors. So, and we could see, here's another figure to this other side where we have stained also for different markers. So we can actually see that the development is the same even when the tumor is in there for the neurons. However, the tumors are growing. We wanted to use this as a platform for drug discovery or for drug screening. So what we did here was we tried 2 different drugs, so TMC and DOX, and these have been promising candidates in vivo in humans. So what we could see is that we saw a decrease in the amount of tumor with both these treatments, and I think it's most visible with the DOX. However, the neurons were not affected, and of course this is important. Of course we can kill all cells in the brain, but then we don't have much left. So it's important that you just target the tumor cells and not the neurons necessarily. So in summary, what I've tried to show you is that there is a lack of effective drugs for neurological disorders, despite tremendous research using animal models. Human models have the potential to overcome species extrapolation. And for this more complex human in vitro cell culture, they are preferable perhaps to model human brain diseases. And in vitro models are highly useful for screening new drugs. And they can be infected by some viruses that also are not able to infect animals or monolayer cultures. But I do believe that we need a combination of in vitro, in silico, and vivo and epidemiology studies to improve the drug discovery. And by the end, I want to finalize with a quote from a statistician or mathematician, George E.P. Box. All models are wrong, but some are useful. And with that, I want to thank you for your attention.
Thank you, Helena. That was super interesting. We are going to wait with questions until we heard Laura's talk. So please type your questions in the Q&A box, and we will have a Q&A session after Laura Gribaldo's talk, which is up next.